Chapter 4, Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to baby elephant, not some other animal? Or why mango seed only forms mango plant, not any other plant? Given that they do, are the offspring identical to their parents? And they do show differences in their some of the characteristics. Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These are several related questions are dealt with scientifically. In a branch of biology known as genetics, the subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed from parent to progeny. It's, it is the basis of heredity. Variation in the degree by progeny differ from their parents. Human knew from as early as 8,000 uh, to 10,000, 1,000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals. So, selectively breed and select for the organism that possess desirable characters. For example, through artificial selection, domestication from ancestral wild cows, we have well-known Indian breeds. Example, Sahiwal cows in Punjab. We must, however, recognize that through our ancestor knew about the inheritance of characters and variation. They had very little idea about scientific basis of their phenomena. 4.1 Mendel Law of Inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on the green peas for 7 years, 1856 to 1863, and proposed the laws of inheritance in living organisms. During Mendel investigation into inheritance pattern, it was the first time that statically analysis and methodologically logic were applied to problem in biology. His experiment had a large sampling size, which gave greater incredibility to the data he collected. Also, the confirmation of his inferences from the experiments on successive generation of these test plants proved that his result pointed to general rules of inheritance rather than being unsubstantiated. unsubstantiated ideas. Mendel investigation characters on green pea plant that were manifested as two opposite traits. Example, tall or dwarf plant. Yellow or green seed. This allowed him to set up basic framework of rules governing inheritance, which were expanded on by later scientists to account for all diverse nature, natural observation, and complexity inherent in them. Mendel conducted such artificial pollination cross pollination experiment using several true breeding pea lines. A true breeding is one of the having undergone continuous or cell pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plants varieties as pairs which were similar except for one character with contrasting trait. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, full or constructed green or yellow pods, and tall and dwarf plants. Figure 4.1, Table 4.1. Contrasting traits studied by Mendel MP. Serial number, characters, contrasting traits. 1. Stem height, tall or dwarf. 2. Flower color, violet and white. 3. Flower position, axial and terminal. 4. Pod shape, inflated and constructed. 5. Pod color, green and yellow. 6. seed shape, round and wrinkle. 7. Seed color, yellow and green. Inheritance of one gene. Let us take example of such a hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plant to study inheritance of one gene. He collected seeds produced as a result of this cross and grew them generate plants of first hybridization generation. This generation is also called filial progeny or the F1. Mendel observed that all F1 progeny plants were tall, like one of its parents. None were dwarf. He made similar observation for other pairs of the trait. He found that F1 always resemble each other, one of the parents, and that trait of other parent was not seen in them. Mendel then self-pollinated the tall F1 plant and to his surprise found that in the filial 2 generation of these some of the offspring were dwarf. The character that was not seen in F1 generation was now expressed. The proportion of plants that were dwarf were one fourth of the F2 plants, while three-fourths of the F2 plants were tall. Tall and dwarf traits were identical to their parents' type and did not show any blending. That is all, offspring were either tall or dwarf. None were in between height. Similar results were observed with the other traits that he studied. Only one of the parental traits were expressed in F1 generation, while at the F2 stage both the traits were expressed in proportion of 3:1. The contrasting trait did not show any blending at either F1 and or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down, unchanged from parents to offspring throughout the gametes. Over successful generation, he called these things as factors. Now we call them as genes. Genes, therefore, are unit of inheritance. Genes, therefore, are unit of inheritance. They contain information that is required to express a particular trait in our organism. Genes which code for a particular contrasting trait are known as allele. IA. They are slightly different form of same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at F1 stage and small alphabet the other trait. For example, in the case of character of height, T is used for trait and small t is used for dwarf. T and T are alleles of each other. Hence, in plants, the pair of alleles for height would be TT, TT, TT. Mendel also proposed that in a true breeding, tall or dwarf pea variety, allelic pair of genes are for height are identical or homozygous, TT and TT, respectively. TT and TT are called genotype of the plant, while the descriptive terms tall and dwarf for the phenotype. What then will be phenotype of a plant that had a genotype TT? As Mendel found that phenotype of F1 heterozygote TT exactly like the TT parent in the parents. He proposed that in a pair dissimilar factor, one dominates the other as in the F1, hence is called dominant factor while the other is called recessive. In this case, T for tallness is dominant over T for dwarfness. That is recessive. He observed identical behavior for all other character trait pairs that he studied. It convenient and logical to use capital and lower case of an alphabetical symbol to resemble this concept of dominance and recessiveness. Do not use capital T for tall and D for dwarf because you will find it difficult to remember whether T and D allele are same gene or character or not. Alleles can be similar in case of homozygous capital T, and small t, and dissimilar as in the case of heterozygous TT, since the TT plant is heterozygous for gene controlling one character height. It is monohybrid and cross between capital T, capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of parental pair separate or segregate from each other. Only one allele is transmitted to the gamete. This segregation of allele is a random process. So, there is a 50% of chance of gamete containing either allele, as has been verified by result of crossings. In this way, the gametes of tall capital T plants have the alleles capital T, and the gametes of dwarf small t plants have the allele small t. During the fertilization of the two alleles, capital T from one parent, say through the pollen, and T from the other parent, then through the egg, are united to produce zygote that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t. Since these hybrids contain alleles which express contrasting, the plants are heterozygous. The zygote of the production of gametes of the plants and formation of the zygote, the F1 and F2 plants can be understood from diagram called Punnett square, as shown in the figure 4.4. It was developed by British geneticist Reginald C. Punnett. It is a graphical representation to calculate probability of possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and left column. All possible combinations are represented in the box below in the squares, which generate square output form. The Punnett square shows the parental tall capital T male and dwarf small t female plant. The gamete produced by them F1 capital T small t progeny, the F1 plants of the genotype capital T small t are self-pollinated. The symbol male and female are used to denote the female eggs and male pollen of F1 generation respectively. The F1 plant of the genotype capital T small t when self-pollinated produces gametes of genotype capital T small t in a equal proportion. When fertilization takes place, genotypes T have 50% or chance of pollinating eggs of genotype T as well as genotype genotype T. As a result of random fertilization, the resultant zygotes can be genotype capital T small t, or small d. From the Punnett square, it is easily seen in one fourth of the random fertilization lead to capital T. One, say, 1 by 2 lead to capital T small t and one fourth to small t. Although F1 have progeny genotype of capital T small t, but phenotypic character seen in tall at FT and TT. Externally, it is possible to distinguish between the plants and the genotypes TT and TT. Hence, within the genotypic pair TT, only one character T is tall as expressed. Hence, character T or 12 tall is said to be dominant over the allele small t or the dwarf character. It is thus due to the dominance of one character over the all of the F1 are tall. Though the genotype is TT, and in the F2, 3/4 of the plants are tall, although genotypically 1 by 2 are capital T small t, and 1/4 are capital T capital T. This leads to phenotypic ratio of 3/4 tall, 1 by 4 TT, and 1 by 2 capital T small t, and 1/4 small t. A ratio is 3 ratio 1, but a genotypic ratio is 1 ratio 2 ratio 1. The 1 by 4 ratio, 1 by 2 ratio, 1 by 4 ratio of capital T small t is mathematically condensable in the form of binomial expression ax plus b by square that has the gametes bearing gametes t or small t an equal frequency of 1 by 2. The expression is expanded as between as given below. 1 by 2t, 1 by 2t square, 1 by 2t plus 1 by 2t x 1 by 2t plus 1 by 2t equal to 1 by 4tt plus 1 by 2tt 1 by plus 1 by 4tt. Mendel self-pollinated F2 plants and found that dwarf plant continue to generate dwarf plant in the F3 and F4 generation. He concluded that the genotype of the dwarf was homozygous TT. What do you think he would have got had he self pollinated a tall F2 plant? From the preceding paragraph, it is clear that though the genotype ratios can be calculated using mathematical probability by simply looking at the phenotype of dominant trait, it is not possible to know the genotype typic composition. That is, for example, whether a tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of tall plant at F2, Mendel crossed the tall plant from F2 with dwarf plant. This he called the test cross. In a typical test cross, an organism pea plant here showing a dominant phenotype and whose genotype is determined is crossed with recessive plant parent instead of self crossing. The progeny of such a cross can easily be analyzed to predict the genotype of test organism. Figure 4.5 shows result of typical test cross where violet color flower W is dominant over white color flower W. Using Punnett square, try find out nature of offspring of the test cross. What ratio did you get? Using genotype of the cross, can you give a general definition for a test cross? Based on his observation on monohybrid cross, Mendel proposed two general rules to consolidate his understanding of inheritance and monohybrid cross. Today, these rules are called principle or law of inheritance. The first law of dominance and second law or law of segregation. Law of dominance. First, characters are controlled by discrete units called factors. Factors occur in pair. and dissimilar pair of factor, one member of the pair dominant or the other is recessive. The law of dominant is used to explain the expression of only one of the parental characters in a monohybrid cross and even an expression of both in the F2. It explains the proportion of 3:1 obtained at the F2. Law of segregation. This law is based on the fact that the alleles do not show any blending and that the both characters are recovered. As such, in the F2 generation, throughout one of these is not seen at the F1 stage. Throughout the parent contain two alleles during the gamete formation. The factor or alleles of the pair segregate from each other and such that gamete only receive one of the two factor. Of course, a homozygous parent produces all gametes that are similar, while heterozygous are produced two gametes having one allele with equal proportion. Incomplete dominance.